Topic 20A, Population Genetics. We'll begin this topic by asking, what is genetic diversity and why is it important to the survival of any particular species? So let's consider the idea that over time, species accumulate differences, and as a result, descendants differ from their ancestors, a phenomenon that had been observed and recognized by biologists long before they understood how or why. Charles Darwin proposed that this descent with modification was the process by which species adapt to meet the pressures of the environment, and in so doing can avoid obsolescence and extinction. Today, this is commonly referred to as biological evolution. So, let's now begin thinking about genetic diversity. And we'll do this by briefly reviewing basic Mendelian ratios. You may recall that in a Mendelian cross, you always start with true breeding parents who produce heterozygous F1 offspring who are then crossed to produce a predictable phenotypic ratio in the F2 generation. This is an artificial scenario that's achieved by carefully controlled breeding and human intervention. You may also recall that the true breeding parents are homozygous for a trait. One is dominant and the other is recessive. This means that the ratio of dominant to recessive alleles in a Mendelian cross is always equal, or 1-to-1, and this is what enables the predictable outcomes. Now, in a real-world wild population of any particular species, the ratio of dominant to recessive alleles for any trait can be anything, and it will rarely be 1-to-1. Let's consider the example of albinism among deer in North America. An allele for albinism will be recessive to the one for pigmentation. Now, if the frequency of these alleles were 1-to-1, we could then predict that about 25% of the deer population would be albino. But we know that this is not true because albino deer are rarely seen. The estimate for albino deer in the wild is about 1 in 30,000, which means only 0.0033% of deer are albino. This tells us that the recessive albino allele is very rare. So, how rare is it? We can figure this out using the Hardy-Weinberg equilibrium. This is an application of binomial theorem that allows us to calculate several things. We can use it to calculate the percentage of alleles that are dominant in a population, the percentage of alleles that are recessive in a population, and if we know those, we can then calculate the percentage of individuals that are homozygous dominant, that are heterozygous, or are homozygous recessive in a population. So we're doing this to calculate and study how genetic diversity occurs within a population. The formula used for the Hardy-Weinberg equilibrium should look familiar. You probably saw it in your first algebra class. To apply it to population genetics or genetic diversity, we're going to redefine some of the parameters, some of the variables. In this case, P is going to equal the frequency of the dominant allele. Q will equal the frequency of the recessive allele. And this means that P squared portion of the formula is the frequency of homozygous dominant individuals within a population. 2PQ is the frequency of heterozygotes in a population, whereas Q squared will give us an estimate of the frequency of homozygous recessive individuals in a population. As an example, let's apply this to a population of 20 individuals. And let's say we know that the allele for red fur color is dominant to the one for blue. And we want to know what the frequency or the proportion of these individuals are carrying a dominant allele and which ones are carrying a recessive allele. So first, we count the number of each color, and we learn that there are 15 red and 5 blue. We know that the blue is recessive, so we can now say that 1/4 of the population is homozygous recessive, or whichever proportion we counted. We cannot do the same for the phenotypically dominant ones because they are a mixture of homozygous dominant and heterozygotes. So to figure this out, we're going to apply the Hardy-Weinberg formula. Now, recall that Q squared represents the proportion of homozygous recessive individuals in a population. So we can pull that expression out of the formula and use it to calculate the frequency of the recessive allele by taking the square root. And we find out that, in this case, the recessive allele is 0.5. Now the dominant allele, represented by P, and the recessive allele, represented by Q, equal all the alleles, so we can say P plus Q equals 1. And then we can subtract Q from P to figure out the frequency of the dominant allele, which in this case is 0.5. Now that we know the frequency of each allele, we can use it to calculate the portion of the population that is homozygous dominant by using the p squared part of the formula. So when we follow through with this, we learn that the frequency of the homozygous dominant is 0.25 or 1/4. We can do the same for the heterozygotes using 2pq, and we learn that the frequency of heterozygotes is 0.5 or 1/2. So in our population of 20 individuals, we can estimate that 1/2 of them are heterozygous and 1/4 homozygous dominant. Now let's apply this to our albino deer example. We already know that the albino deer make up 0.0033% of the population, or expressed as a proportion, there's 4 zeros after the decimal. This means that 99.9967% of the population will be either homozygous dominant or heterozygous. But which proportion is which? So we can use the homozygous recessive portion of the population, which we know, and use it to calculate the frequency of the recessive allele. So when we take the square root of the proportion, we find out that 0.0057445 is the frequency of the recessive allele in the population. If we subtract this from 1, we find out that the dominant allele makes up 0.9942555 proportion of the population. Now we know the frequency, or the proportion, of both the dominant and the recessive alleles, and we can use that to calculate the homozygous dominant by using p squared, and we find out that, or we estimate, that the homozygous dominant is about 98.85% of the population. Now we calculate the heterozygote, and we learned that heterozygotes, or carriers for albinism, make up about 1.1% of the deer population. To review, genetic diversity allows a population of any species to avoid extinction. We can calculate simple phenotypic and genotypic ratios using the Hardy-Weinberg equilibrium.